So hello everyone, my name is Balázs Gyuris. I'm a PhD student at the University of ELTE at the Department of Genetics, and I also work here in this building in the Institute of Archaeogenomics as an archaeogeneticist. And today I will talk about a site of Kölket-Fekete-kapu, following the footsteps of Tamás, but now we will integrate ancient DNA analysis too. This analysis of this Kölket-Fekete-kapu site is part of the Histogenes project. So as a geneticist, I wanted to give a brief introduction about the connection between genetics, ancient DNA, and children. So what do we know? What are the possibilities? What are the methodologies? And what studies we can find actually children? So, for example, I presented two papers here from the Neolithic period. On the left, this paper is from France and analyzed a Neolithic community and reconstructed a very nice, huge pedigree. And after that, the authors tried to measure the grave distances, the connection between the grave distances and the connection between the grave distances and the connection of the relatedness. So, for example, in the case of subadults, that if in first-degree connections, like in father-son connection, is there a closer connection to the father sons or uncles? So they tested this, which is a very nice paper in that sense, and also staying in the Neolithic times. There is another paper analyzing a Neolithic tomb from England, and they also had a huge, nice pedigree, and they actually interesting part of the study that they suggested that there were a possibility of adopted children in this community. So as we see it in most of the cases in archaeogenetics, in children are connected through these pedigrees. So there are a lot of other ancient DNA studies, for example, which are analyzing, doing transect studies, analyzing a region or a period, but their children are data points, so they are not subject of the analysis. So, but in these pedigrees, we can tell more about true social organization about children. And there are also more biological-oriented methodologies and directions. For example, a study from the Max Planck Institution where they wanted to find traces of trisomy, and they analyzed a huge ancient DNA dataset with thousands and thousands of samples, and they found a few cases in where they were able to find traces of trisomy in children, traces of Down syndrome or Edwards syndrome. Or another very interesting method that we can actually use now with our ancient DNA samples is measuring the possible parental relatedness between individuals. So it can be a very good sign. So these long rows of homozygotic segments can leave these special genomic traces in the human genomes, and we can actually detect close relatedness. For example, in this figure, a cousin relatedness can appear and leave its traces in the genome. So the presence of these row segments can be very informative, and also it can be a very good future direction for biological analysis too. So I'm a really fortunate situation because Tamás has already introduced the Kölket-Fekete-kapu site. So just for few sentences, so the Kölket-Fekete-kapu site is located in southern Transdanubia region. In the earlier period of the site, we see a very diverse material culture with Western European and also late antique influences, while through the middle of our period and in this transitional phase, we see the arrival of newcomer communities in huge grave groups, and we see an overall impoverishment, overall homogenization process through the site. And in the Histogenes project, we had the possibility to analyze 360 ancient DNA samples from the site, which is a lot, but actually the site itself is huge, with more than 1,300 graves. And also we have integrated isotopic samples in our analysis too. So as a before talking about the children of Kölked, I wanted to give a brief introduction about the genetic results to see the general trends in the site. Here in this plot, we can see a principal component analysis with the background with gray dots, which is probably, you cannot see it, but there are gray dots in the background of this BCA plot. I will find this laser. Yes, it's here. And thanks. And these gray dots are modern individuals from Eurasia, and we are projecting our ancient individuals into these modern individuals, and we can have beautiful gradients there of ancestry. So on this side of the plot, we have Northern Europe, and we can go down to Southern Europe, and this part of the plot is Eastern Asia. And with blue, there are other early Avar period individuals, mostly from the Danube-Tisza interflow region. And what we can see that there is a shift in ancestry, so there are lots of individuals with this Eastern Asian ancestry. But in the case with the Kölked Fekete-kapu individuals with this purple color, we can see that most of them are this European diversity. So the appearance, there is no Eastern Asian ancestry in high number in the early period of the site, but when we move to the late period of the site, we see the appearance of this Eastern Asian ancestry in high number. You will see in later slides that where does it appears, and so in this transitional phase, this Eastern Asian ancestry appears in the site. So just for a very, very short introduction about the pedigrees and the site and what are the major takeaways between the periods and the pedigrees. So here you can see the cemetery map in these parts. You can see the A cemetery part of Kölked with a huge early period pedigree. And these in the upper part, you can see the separated grave groups in the cemetery, most related to the later period of the sites, and some early period grave groups are also appearing here. What is the most important takeaway here is that we have these separated grave groups, and the biological relatedness is very low between these grave groups, so we don't see any kind of connection between them. We don't see the actual connection between the pedigrees, and there is a minimal continuity from the early period to the late period in the cemetery. So, and what is also very important that in these grave groups there are different differences in material culture too. So we have a very nice picture here about these differences, and based on Norbert Faragó analysis, I wanted to show just one side about the strontium isotope analysis, what we are also incorporating now into our study. Also, you can see the separation of the early and the late phase of the site here with these yellow and the orange colors with sobadotes, and with the red and the blue colors, with red colors females, and with blue color males. And what we can see that in the early period of the phase, we have a high number of females appearing probably from other regions. There is a sign of mobility in this case. They are out of the local ranges, and they are out of the ranges of the subadults from the early period. While we have a more homogenic picture in the males, but we have outliers in every direction in this plot. While in the late phase, we can see a little bit of switch of the baseline, but not a huge one. They are all in the local ranges. These non-adults are in the local ranges also in the late phase, with a few exceptions, for example, with the two individuals which will be important a later part of my presentation. That about the females and the males had a show pointing probably the other direction, and there is a probable mobility in the case of the later phase of the site. So now turning to the pedigrees, you have seen lots of pedigrees today, so I won't show that many, just a few which the important and interesting one, and also which are related to children and some new results. So if you want to understand the major elements, major trends and we want to connect with our pedigrees and genetic results, we had to see, we had to understand the major archaeological trends in this site. So in this bottom half part, you can see an analysis made by Zsuzsanna Hajnal, who is doing the archaeological investigation in this study, and the differentiation of the different grave goods types between subadults and adults in the different periods. So as it's, I think, impossible to see the most of the details on these in the small figure, I will just drive you through it. So what we can see in the early period, that late antique traditions with, for example, with this blue and also with red colors, and the traditions with pointing to Western Europe are there in a high number in all the subadults and also in the adults' grave. Why the green means that there is no grave goods in that sense. And we can see that in the late period, these type of grave goods disappears. You can see the green colors also in the subadults and also in the in the adult graves. So this pedigree is important because it reflects this diversity of material culture, so late antique elements and also this Western Europe, grave goods pointing to Western Europe are also appearing in this pedigree and also in males, females. So it's a very fascinating one. And also in the first generation, we see appearance we see the first generation, the founders of this pedigree, they are outliers also in the strontium analysis. And you can see an European PCA, and you can see that they are all in this European diversity. But it was really fascinating that we had this young girl grave who is between the age of 11 and 14, and this girl had a complete adult female set with earrings and also with bed necklaces, maybe referring to high status of her. And also it is important to say that there were other few graves, not that much, but a few graves with female sets in the cemetery. But here we can actually connect it to this pedigree, and we have other early period pedigrees where we can see this actual diverse material appearance of this diverse material culture, and they are also in this European diversity, as you can see it on this PCA. And what was very interesting in this early period pedigree, that as you can see here in this graph, the different age groups are there with these with the blue, red, and the green colors, and the purple are the adults. And what we can see that in the younger generations, it's from the whole cemetery. In the case of the younger generation, we can see, for example, these Meroving belt sets, but the other belt sets are not appearing in the youngest generations. And also we can see that different types of weapons are not appearing in the youngest generation in the early period, but arrows and arrowheads are appearing in this in the whole site in the youngest generation. In the case of this pedigree, we can detect, we were able to detect that that some of this high status male had a full set of adult material, in that sense that he had a middle avar belt and also arrowheads in his grave. So maybe he's a successor. They wanted to be a successor in this community. So this is also very fascinating. Showing this very fascinating richness of the early period and also the child graves. And moving to the late period groups, so in the late period, as I mentioned, the important genetic takeaway is that this Eastern Asian ancestry starting to appear, and also we see this impoverishment process. We see that the large grave groups appearing the surface of the base cemetery, and what we see in the graves that the number of Avar belts in the adult among the adults was high, but we don't see it in the case of children. While in the case the animal skin, it appears in some cases also in the younger generation, but in the adults there are a lot of examples for animal skin. And when we are moving to earrings or beads, it's appearing also in higher number in the younger generation. So when we compare, if you bear in mind these figures and also the first slides that I have shown about ancestry and the change in ancestry, we can actually tested for analyze these different grave groups, and what we have seen that there were late period grave groups. For example, this grave group 1, we have these smaller pedigrees here, but the individuals had no Eastern Asian ancestry, so it was very this was the only grave group in the later period where we see no Eastern Asian ancestry. Also genetically they were in this European diversity. There were signs of possible continuity based on uniparental data. And what was fascinating that the overall impoverishment process is also appearing in this pedigree. So regardless the ancestry differences to the other late groups, but we found that that the young age children, in the case of these females, had these beads, while the others had, adults also had no grave goods. While, as I mentioned, in the case of these other of our late period groups, like this group 12, you can see here in this PCA plot that compared to this grave group 1, the appearance of Eastern Asian ancestry is there. So there are probably there are also signs of newcomer females in this community, and we can see that actually also in adults, but also in children, there is a differences. For example, in this case, the animal skin appearing in the younger generation too. And as for one closing slide, we had a very interesting case study in the study about grave group 13, which was very different from all these grave groups in every aspect. So basically, we have seen that in grave groups they were poor. But what was very fascinating that generally in the late period of the sites, we see that the connectedness is very high. So we see not just first, second degree connection and these large extended pedigrees, but connection where we cannot reconstruct the actual pedigree, but we see that there is a biological relatedness between individuals. So they were very, very connected. But in the case of this grave group, we have found that there were only 2 first degree connections, but other than that, there were no connection at all up to the 3rd degree. And this was very rare in that sense. And the other additional thing was that in ancestry, as you can see here in this PCA, they were very diverse. So there were like individuals with Northern European ancestry pointing to Southern Europe, also the Eastern Asian ancestry appearing in this community. And to our surprise, we have found that actually in strontium results, they were outliers. So we have these 4 individuals analyzed in the strontium values too. And the 2 outliers were, we have found 2 outliers. Those were the only outliers, subadult outliers in the community. So if wrap up things, we don't know what's going on here, but it's very fascinating. And as a conclusion, in conclusion about ancient DNA and how we can connect it with children, so through the reconstruction, through this pedigree, we can actually say something about social organization, and actually this can allow us to understand the role of children, and also it's very important. The ancient genomes hold lots of valuable information that that maybe in the future can be a good direction for the analysis of children. So there is a lot of things to do in that sense. And in conclusion about the Kölket-Fekete kapu site, so we see this in the early period. We can see this high diversity in archaeological material and also in the European genetic variation too. And we have found these very rich graves of children too. And we, in the later period of the site, we see these cultural and genetic differences, which was reflected also in the child graves. And I would like to thank you for your attention, and I would like to thank you all the co-authors and the Histogenes project itself.